Hi, this is Dr. Grace Klinges, a postdoctoral researcher here at Mote. So Grace, can you tell me about your background and the focus of your research here at Mote?
Sure. I got my PhD in microbiology from Oregon State University just last year, and I started at Mote in October, so I'm one of the newest hires here. And my research focuses on bacterial parasites and pathogens that are involved in coral disease.
Cool. What made you decide to study corals and make it your career?
So I grew up on the Jersey Shore, living in the ocean, spending a lot of time underwater, and I became an avid scuba diver. But I also had a lot of interests in microbiology. So it just made sense when I went into graduate school to pursue a career where I could do both microbiology and get to scuba dive all the time.
What is the Coral Health and Disease Program?
The Coral Health and Disease Program, the program that I'm part of, studies both the environmental and genetic factors that cause coral disease and other types of coral stress, as well as the bacteria and viral pathogens themselves that cause disease. Dr. Courtney Claypack, who's the other postdoctoral researcher in my program, she studies more of the resilience aspect of coral health. So studying how corals respond to stressors such as thermal stress caused by ocean warming. Whereas I study coral disease, which is a huge problem in the Caribbean.
Cool. So based on your research, what are the most common coral diseases that impact corals around the world and in Florida?
Well, historically, the Caribbean has been impacted by diseases such as white band disease, white pox disease, black band disease, and yellow band. And these diseases are named kind of, as you can tell, they're just named based on how they look on the coral. So just morphologically. But recently, starting in 2014, a disease called stony coral tissue loss disease cropped up starting around Miami. And since then, it's spread throughout the Florida Reef Tract and throughout the Caribbean. And that's caused massive mortality across 20 different species of coral.
Okay. So how prevalent are these diseases right now?
So stony coral tissue loss disease has spread aggressively, but those other diseases I mentioned, they occur seasonally. So we will see occurrences of each of those diseases during the summer, during the warmest months of the year, but they're usually pretty isolated.
Are these diseases caused by climate change effects such as warming or higher acidity in the oceans, or are there other factors as well?
They're not directly caused by environmental stressors. We know that most coral diseases we believe they have a bacterial or viral role in them because they can be transmitted from one coral to another, and also because they can be treated by antibiotics. But a lot of environmental stressors can make coral diseases worse. In particular, we know that nutrient pollution and thermal stress both exacerbate coral disease. And actually, overfishing, which you wouldn't really think would have a role on coral disease itself, can make coral disease worse because it allows for the growth of macroalgae, which covers the coral and allows the coral microbiome to shift dramatically. And that can stress out the coral and make it much more susceptible to disease.
Oh, wow. So how do you make corals more disease resistant?
Well, this is actually a project that we work on with the coral reproduction and restoration programs. So we can look at the genetic variability of corals and assess how their disease susceptibility varies. But we work with these other programs to make babies that are better at responding to disease. So if we can identify a coral that's better at responding to disease, we can select that as a parent to use in assisted sexual reproduction activities. But we can also look at the microbiomes of our corals that we have on site and make predictions about whether they might be disease susceptible or disease resistant in the field.
So what is the most interesting discovery you've made here at Mote?
So I actually did a lot of my PhD research here at Mote. So what's been amazing is, as a postdoctoral scholar, I've been able to continue some of those projects. And one of the most exciting things that's happened since I've been here as staff is we actually identified some of the early life stages, the microbiome changes in early life stages of coral. And one of the things that was interesting to me is this parasite that I've been studying, and I studied during my PhD, is a common parasite of staghorn coral and causes a lot of impacts on coral health. And we thought that it would be passed from parent to spawn when corals reproduce, and we found that it's actually not. It must be inherited from something in the environment. So one of the things we're working on now is to figure out what that vector might be. So if the corals are picking up this nasty bug from the environment, what could be giving it to them? Is it from other coral? Is it from something like a snail or a fish that's grazing on the coral? Or is it just from the water surrounding the coral?
Okay. Are there any projects that you're doing now or looking forward to doing in the future?
Yeah, absolutely. One of the projects that I'm looking forward to continuing that we started late last year is actually a stony coral tissue loss disease study. So that disease I mentioned is becoming really prevalent and problematic. we were able to get out to Dry Tortugas National Park, which is a very remote national park west of Key West. And we were able to sample 36 colonies in advance of the disease wave on Loggerhead Reef. And we're going to return to those colonies when they inevitably become diseased. And we can look at genetic variability of those corals and see if there's genetic reasons why they are resistant or prone to disease. And we can also look at the microbiome shifts, the bacterial and viral species that change throughout disease progression, which might give us some more information about what causes that disease, which is obviously really important for solving it and treating that disease.
Cool. How do you guys determine which corals are disease resistant?
So we can actually use experimental assays in the lab to expose corals to known pathogens that cause coral disease. And that way we can see which of our genotypes or different genetic variants that we have on site might do better when exposed to disease or might get sick really easily. Why don't we go upstairs and we can check out some of the ways that we assess disease on site.
Sounds amazing. Let's do it. I'm here.
Hi, Maddie.
Hello.
Let's look at some plates.
Okay. Oh wow, this is cool.
So here we've got Serratia marcescens, which is a known pathogen that causes white pox disease in a Acropora palmata or elkhorn coral. It's nice and pink. Sometimes you'll actually see this on your shower curtain. If you see your shower curtain turn pink, yep, that's Serratia marcescens. It's not pathogenic to humans, but it will cause disease in corals. And actually this strain was isolated from human sewage. So we found probatively that human sewage causes white pox disease in elkhorn coral. So what we can do with this pathogen that we can culture on site here is we can take a swab of this pathogen and grow it up in some sediment from our coral nursery, so just sand. And the sand will become coated with the bacteria. It'll grow all over the sand, and then we'll sprinkle the sand on a coral fragment. And if the coral gets sick, we'll know it's disease susceptible. If the coral doesn't get sick, then we can explore more and see if it's disease resistant.
Okay.
Here's some other coral pathogens that we work with on site. This is Vibrio. This is the genus Vibrio. I don't know what species this is because it's a very common coral pathogen. And we found this on our corals just on site. And it's not likely to kill the coral, but it's a sign of coral stress. So we are exploring different ways that we can get rid of the Vibrio in our corals. So whether it's antibiotic treatments, or actually we can look at bacterial predators that eat Vibrio. So one species is called Halobacteriovirax. It's like it sounds like a dinosaur almost. And Halobacteriovirax will eat Vibrio species and remove them from the coral microbiome. But the Halobacteriovirax themselves are not harmful to the coral. And Vibrio can look like lots of different things. This is also a Vibrio species. But we can grow all of these here safely on site, and we can conduct experiments using these pathogens to understand disease susceptibility in our corals.
That's so cool. Hello.
It's going to be really long.
Anything that's going to be small is going to be huge on me, just as my size. Ready.
Slippery. Be careful. You'll write your name on it.
Okay. My name?
Yeah.
Just like right here?
Wherever you want it. It's your plate.
First and last, or just first?
First is fine.
Okay.
I don't think we have any other Maddies.
Yeah.
We're going to make a Serratia plate today. Okay. Is this one not open? That's why. No. It's just open on the wrong side. Okay, so what we're going to do is we're going to take one of our existing plates.
Okay.
And I'm going to sterilize this loop. I'm going to heat it up until it turns orange, and then we're going to use that sterile inoculating loop, that's what this is called, to take bacteria from this plate and add it onto our new plates.
Okay.
So I want to see it start to turn a little bit orange. All right, there it goes. So now we've killed everything that's on this loop.
Okay.
Want to wait a few seconds, let it cool. And then we're going to swab from our plate. And I'm going to make a pattern. I'm going to spell my name. My initials. And that's it.
That's so cool.
Now your turn. You can do whatever pattern you want.
Okay.
And the bacteria will grow in that shape. All right, your turn.
Second?
Mm-hmm. Oh, other way around.
Oh, other way around?
Yep.
Okay.
And now just do whatever you want with it.
There we go. It's a little
Nice
wonky,
job.
but it works.
In there. Cool. And now we'll just
Thank you.
put this in the sink. And now we can get your plate in the incubator and see what it looks tomorrow.
Thank you.
My name on this.
That was so cool.
Nice work.
Thank you.
Cool.